Good afternoon, everyone. This is Jonathan Maudsley. I'm the Science Advisor for the Association of Fish and Wildlife Agencies. I'd like to welcome you to this afternoon's webinar, which is being presented in partnership between the Association of Fish and Wildlife Agencies and the U.S. Forest Service, specifically the Research and Development group within the Forest Service. Today we're going to be learning about the National Genomics Center for Wildlife and Fish Conservation. Our presenter will be Dr. Michael Schwartz and some of his colleagues at the Rocky Mountain Research Station working with that center. And would like to, before we begin, like to go over some brief housekeeping rules that we have. First, if you are not the person speaking, please mute your phone on your end. We ask this out of courtesy so that we don't hear people typing or eating lunch or whatever it is you happen to be doing. If necessary, we will get on the phone and mute all of the lines except for the speaker, but we'll try it first with just folks muting their individual lines. And then this webinar presentation today is being recorded. We will make the recording available via the Association's YouTube channel, the Association of Fish and Wildlife Agencies. And we appreciate your opportunity to make this recording available so this information can be more widely distributed. Finally, if you do have a question for the speaker during today's presentation and we have time for it, we will take questions at the end of the presentation. If you want, there is a chat function in the lower left-hand corner of your screen which you can use to ask the speaker a question. We'll start at the end, if we do have time for questions and answers, with those questions that people have typed into the chat box. So please go ahead and feel free to present your questions there. And so I'd like to officially welcome you on behalf of the Association of Fish and Wildlife Agencies. This is a topic that we know is of great interest to many state fish and wildlife biologists and to many of our agency folks, and so we're really delighted to be co-presenting this webinar. And this is a part of a series that we host with Forest Service R&D to try to bring good scientific information to the attention of the states. At this point, I'd like to turn it over to our colleague and co-host, Dr. John Roethlisberger, to introduce the speaker and provide an introduction on behalf of the Forest Service. John.
Thank you, Jonathan. And on behalf of the Forest Service, I would like to welcome everyone to today's webinar. Thank you for calling in. As Jonathan mentioned, this is John Roethlisberger. I'm the National Program Leader for Fish and Aquatic Ecology Research for the Forest Service. And the Forest Service appreciates this opportunity to partner with AFWA to provide this webinar series. Our goal is to share the latest in scientific research and synthesis that is relevant to natural resource policy and management. Today's speaker is Dr. Michael Schwartz. Dr. Schwartz is the Director of the National Genomics Center for Wildlife and Fish Conservation. The center is based in Missoula, Montana, and it is a multi-agency resource which provides defensible, reliable, and cost-effective genetic and genomic research and monitoring solutions to U.S. Forest Service and its partners. Mike was recently honored by Thomson Reuters as one of the world's most influential scientific minds. And I think we may have some folks out there that aren't on mute yet, so if you're not...
Yes, please mute your phones if you haven't already done so.
And Mike was also named as one of the most highly cited researchers in 2015 by Thomson Reuters. Two of the staff from the National Genomics Center will also be speaking today. Those individuals are Tommy Franklin, who is the center's environmental DNA coordinator, and Christy Pilgrim, who is the center's lab manager. And I will let Mike introduce them a bit more later on, but for now I will go ahead and turn the time over to him.
Yeah, and before Mike starts, this is Jonathan Maudsley at AFWA. We're going to mute the lines and turn it over to Mike and unmute his. So hold on 1 second.
The conference has been muted.
Okay, Mike, you're on.
All right. Thank you. Thank you very much for hosting this seminar. I just want to take a moment and thank AFWA and Jonathan Maudsley and John Roethlisberger. I really appreciate you giving us the opportunity today to speak about our relatively new National Genomics Center for Wildlife and Fish Conservation and things that we're trying to do to provide natural resource to provide DNA data for making natural resource decisions, whether it's on National Forest Service land or whether it's with our state partners. All right, next slide. So the topics today, I'm going to provide some background on the National Genomics Center, and I'm going to talk about this new model of collaboration that we're experimenting with. We're then going to move into some what we like to call conservation solutions at large scales, and we're going to talk about some of our connectivity work, especially as it relates to some projects that involve non-invasive genetic sampling. We'll then turn to another example of conservation solutions at large scales, and that's where we think we have our strongest niche, and that's in our environmental DNA program. And hopefully many of you on the line have partnered with us or worked with us in our eDNA program. And lastly, if we have time, I'll try to pull it all together, and I want to talk about how we're moving from the DNA lab all the way to the iPad. So how we're developing tools, disseminating results, and getting them on the ground to be usable to folks in both federal and state agencies. Next slide. Okay, today's speakers, I'll start off giving a little bit of the background, but I'll spare you from having to listen to me for 45 minutes. I'll turn it over to Christy Pilgrim, who's our lab manager, our overall lab manager, and she's the leader of our non-invasive and forensic projects. She'll speak on the next section, and then I'll have Tommy Franklin, who's recently become our eDNA coordinator when Kelly Kareem, who was our eDNA coordinator, left and is now working in a tribal liaison position between the National Genomics Center and the many tribal projects that we have ongoing. Next slide. Now, I would be remiss if I didn't thank a lot of other folks whose data I'm using. I couldn't put everyone's picture up. We work with so many people. Whenever you do a large, broad-scale project, you're going to involve a lot of different people. But I do need to mention these folks here I put up on the top who have been instrumental in the center and its success and its direction. And that's Kevin McKelvey on the left, Kelly Kareem, Dan Isaac, who will talk about some of his data and models, and Mike Young on the right. Next slide. Okay, so we started off as many folks do, fairly small. We were interested in really just one species at the time. About in the late 90s, we realized that almost every wildlife project was beginning to have a genetics component. And so we started moving into the realm of genetics in the late 90s, and we really got involved with non-invasive genetic sampling with, in particular, one species. Since that time, we've grown our partnerships and our collaborations and our projects almost exponentially. And so we decided we needed to start to change our model. Next slide. And sorry, I should say, while when we started the lab, before we changed our model, we really were trying to adhere to several principles. And that is, we felt we could best serve our state agencies and best serve our federal agencies if we could provide them defensible, reliable, and cost-effective research and monitoring. And John Roethlisberger had mentioned that these were our core principles early on, and I think these core principles have served us well. And by these principles, I mean defensible, we're trying to publish all of our data. Reliable, we're trying to at least quantify genotyping error rates and other rates, and show how data can and shouldn't be used. And cost-effective, we're trying to always move to the most cost-effective solutions that may not always be the cutting edge. Sometimes they are the cutting edge, but ones that are cost-effective, we can get on the ground. Okay, next slide. So, we started it, like I said, in 1998. We restructured, holding those core principles in 2014. And we basically were trying to move away from just becoming a service lab or just a research lab or one where our partners would fund us to take on a particular project. We realized that almost all of our large-scale projects, and many of even the smaller-scale projects, were multi-agency partnerships. And so we felt like the right solution for the future is going to be when our partners, at whether it's a state or federal or tribal level, come work with us, train with us, help develop the questions with us early on, and work with us through the entire process from inception of the project through implementation. And so we're trying to set up our new center in such a way that it's a large-scale multi-agency partnership. Key to that has been setting up MOUs. We have an MOU with the University of Montana, and that's to share equipment. And that's just because genomic equipment is really expensive and it goes out of date fairly quickly. So we can offset a lot of those costs with that kind of an MOU. We also have signed an MOU with the Fish and Wildlife Service. Next slide, please. And that MOU with the Fish and Wildlife Service is allowing us to meet the goals that we've established in our charter. And I put this quote up from our charter, really, which I think captures the direction that we're trying to go with this new model of collaboration. And that is, our vision is to gain synergy in our management decisions and cost effectiveness in our research by hosting and co-locating individuals from multiple agencies with different missions but similar information needs. And really, this has been driven home to me several times in the last couple of years when we've done some of our eDNA sampling, which Tommy Franklin will talk about later. But we've actually run across a couple cases where people from either different state agencies or a state agency and a federal agency or two federal agencies will actually be sending us data from pretty much the same point because they want the same information for the same species at the same point. And they don't know that each other is sampling that location. So we see a lot of cases where we can gain synergies, be more cost effective by functioning in a multi-agency way. And that's how we're trying to design this center. Next slide. Before, as I mentioned, our way to do this is through the MOU. I mentioned the MOU with the University of Montana. Here's the example of the MOU with the Fish and Wildlife Service, and it's been endorsed and was signed by both Dan Ash, the director of the Fish and Wildlife Service, and Tom Tidwell, the chief of the U.S. Forest Service. So again, they both believe in the mission of trying to gain these synergies across broad scales. Next slide. The focus of the center, the next few slides I want to show you what I see as the focus of our center. There are a lot of good genetics labs out there, a lot of people doing really exciting work. There's a couple things that we're going to focus on, and I'll talk about our niche in a second here. But really our focus has kind of a tripartite mission. Number one, we're going to stay with our non-invasive genetic sampling and connectivity research, and connectivity and substructure research, which is basically where we cut our teeth back in the early 90s. We've been doing non-invasive genetic sampling, working with low-quality DNA samples for a long time. We're staying with that. Recently, in the last 5 years, we've moved to environmental, 4 or 5 years, moved to environmental DNA work. We have a very large environmental DNA program. We'll talk about that. And we also, obviously, being a genomics center, we do some genomics work. But really what we're trying to do is mix our genomics work with our environmental DNA work and with our connectivity research. Next slide. Projects, just to give you some sense of the scope of projects. We are located in Missoula, Montana, and that's really, there's a lot of federal lands out here, so you can see we have a lot of projects out in this part of the world. I'd like to say this is all of our projects. It's not, but we put our slides, I put these stars on the map just to get a sense of where we have projects and that we are bigger than just something in the Rocky Mountain region, even though that is where we end up being somewhat focused. We also have several international collaborations as well. Next slide. And in terms of species, I'm sure some of you out there are wondering what species we work on. Some of you know us for maybe one particular area. We often find that when we talk to a collaborator, they either know us because of our eDNA work, or they know us from our lynx work, or our fisher work, or something like that, or wolverine work. I just wanted to I couldn't put all the species. I think we work on 60 different species right now. The main ones that we are working on are more recent projects I've listed here, or some that people might not have known about. Here are some of the carnivores listed on the left. We still have a large database and large projects on lynx and mountain lions, martens, fishers, wolverine, and some wolf projects. We do some small mammal work, some ungulate and bovid work. Next slide. But we also have had several projects on birds, and at some point I would love to share with you, maybe in a joint presentation with some of our other collaborators, some of our sage grouse research. We have an enormous sage grouse project that's spanning 5 years, that's covering entire geographic distribution of sage grouse. But we have large projects on birds, some amphibian work, some invertebrate work, and then of course with the eDNA component coming in recently, fish have taken a larger role in the center. Next slide, please. But really, as I mentioned before, there are a lot of good genetics labs, a lot of good research labs. I think our niche and our specialty, where we can maybe do the most good, is when we try to find conservation solutions across broad scales. And I put these two images up here because this is where we have succeeded in the past. We have a paper here on the left on wolverine, where we've been able to work with the fact that we have an agency that covers 193 million acres of land and has districts all throughout. So we actually have almost a small army of people that can go out and put samples, or can sample or do non-invasive sampling on the ground across a broad area. And we can work with partners to fill in other locations. So on the left, we can talk about some work that we did with Idaho Fish and Game, Montana Fish, Wildlife and Parks, some of the national parks, to understand wolverine connectivity. Again, looking at these broad scale questions. Where do we have connectivity across broad scales? On the right is our example for sage grouse. You can see the sampling distribution of sage grouse, and that's been, I think 11 different state agencies have been partnering with us to work on that project. So again, our specialized niche is really this conservation solutions at broad scales. Next slide. Okay, so the first example, and I think we can best show this by examples. The first example I'd like to share with you today about one of these conservation solutions at large scales is one of the databases that we've accumulated over time. It was one that we didn't initially start out working with, but it was a study that initially started at a small scale and then grew into a fairly large database. Is a project on mountain lions or cougars that I'll let Christy Pilgrim, who I'll introduce now. She's been our lab manager and leader of our non-invasive projects for about the last 15 years. And so she'll be presenting the first case study that we have on mountain lions and connectivity across broad scales. So Christy?
Thanks, Mike. Next slide, please. All right, so what I'd like to do now is use cougars, as Mike mentioned, as a case study to help illustrate the services that the National Genomics Center has provided to our multiple state partners with respect to both non-invasive samples, and in this context I'm primarily talking about hair and scat samples, as well as the utility of having these large landscape scale DNA databases available. Next slide. So this work would not be possible without numerous state partners, and I just have a snapshot of the ones that we've worked with just in particular on cougars. We have other state partners that have been involved with research with other of our species, but we've done a fair amount of work with our state partners on cougars. Next slide. So I just want to introduce you a little bit to this species if you're not familiar. So cougars, also known as mountain lions or panthers, they are one of the widely distributed mammals in the Americas. They're a generalist, found in every major habitat type in the U.S. And while they are a strict carnivore, they're very flexible in their diet. So I say there they'll eat everything from insects to ungulates. They eat armadillos in Texas, for example. And not surprising, they are capable of very long distance dispersal. Next slide. So this map shows the distribution of cougars. I'm going to use this a couple times in my talk. So in green, you can see the distribution of established breeding populations, so largely throughout the western U.S., as well as the Florida panther population, which is an endangered, listed endangered species. And then there's three relatively new established populations. These were naturally recolonized in North Dakota, South Dakota, and Nebraska that all happened within the last 15, 20 years or so. And then in the middle part of the U.S., you can see all of those dots, and as well as in the east. And those dots represent confirmed cougar sightings over the last 16 years or so. And many of those we have in our database because those were sent to us as non-invasive samples for largely hares and scats. But you can see that there's a lot of interest, if you just look at this map, where there are cougars that are now showing up outside of established breeding populations. Next slide. And our DNA database has really focused on trying to capture the populations that are in the easternmost distribution of their habitat, so Montana, Wyoming, down through Texas, as well as the Florida panther, so really, and the populations in North, South Dakota, and Nebraska, so that we have these reference samples in order to help inform. And at the moment, we have a database that comprises over 3,000 unique individuals. Next slide. And the reason this is important is that as these animals continue their expansion and dispersal events eastward and are showing up in places like Ohio and Iowa and Missouri, land managers want to know where the animals are coming from. In addition, one of our big services to our state partners has been using hair and scat that is collected after potential cougar sightings in order to potentially confirm through DNA evidence the presence of these animals in new landscapes and new habitats. And as we mentioned before, these DNA databases can help track these animals across the landscape. Next slide. So I put this slide in here. This is from a radio telemetry study of the Black Hills cougar population, just showing movement of subadult males. And so what the researchers found, in the course of a 3-year study, they didn't have a single subadult male that stayed put, that was recruited into the population. And again, this just kind of gives you a sense of scale on a few of the individuals and their movement capabilities. Next slide. So again, these large DNA databases can help assign population of origin. So what you're looking at is a map of a subset of our samples in our database plotted on the landscape. And each point represents a unique individual, and the colors are part of or help inform population structure. So for example, if you look in North Dakota, largely the cougars that we've sampled in that state all appear blue. So they have their own unique genetic signature. And we can help use that if you look at, there's a blue point in Minnesota, so we can help managers say the most likely population that that individual came from is probably North Dakota. And incidentally, I'd like to point out that red blob of points that encompasses the Black Hills area of South Dakota, eastern Wyoming, as well as the Nebraska population, which was established from animals from South Dakota. And then you can kind of see in the Midwest a lot of red dots. So that's seeming to be quite a source for a lot of these individuals. Next slide. So I'd like to tell you a little story as we're thinking about these animals across the landscape. So in June of 2011, a cougar was hit by a car on the Milford Parkway outside Milford, Connecticut. And as you probably can understand, this is largely outside of any established breeding populations. Next slide. They were able to do a necropsy on this individual. They determined that he was a young male, so 2 to 3 years old. He didn't have any tattoos or collar marks or any indication that he had been a pet or had been in captivity. And he had porcupine quills stuck in his cheeks, so he had at least eaten a porcupine before his death. And the managers in Connecticut, this was a very big and surprising occurrence for them, wanted to see if we could help inform maybe where this animal came from. So they were able to take a tissue sample from this individual, next slide, and sent it to us. And indeed, genetically, that young male most closely assigned to the cougars in that Black Hills region as well as Nebraska, sort of that breeding population. Next slide. But what was even more exciting was that we had some additional DNA results on this particular cougar. Next slide. In that this individual was already in our DNA database. So what this picture is a photo from a trail camera from a year and a half before in Wisconsin. And land managers in Wisconsin started getting reports during this time of a cougar in the area, and they were able to go and collect hair and scat. And we were able to genotype this individual, get a unique ID. Next slide. And so in fact, the genetic analysis, the match probability was 1 in about 800 trillion chance that they just matched, their signatures matched by chance alone. So very strong evidence that this is in fact indeed the same individual, and you can actually see in both photos some of the young spots that this male had, still kind of a young guy. Next slide. And this is a map put together just kind of showing sort of a trajectory perhaps of this animal. So again, genetically he most closely assigns to that breeding population of the Dakotas and Nebraska and Wyoming. First sighting that we had confirmed hair evidence from was 2009 in Minnesota. He moved all around Wisconsin for a couple months. Last sighting was in Michigan in May of 2010, and then a year later he was hit by a car in Connecticut. So this kind of illustrates the ability, again, a huge landscape scale, that by having these samples and all of our partnerships with all the various state agencies, that we can grow this database to be able to help inform when an event like this happens. Next slide. And this is an ongoing research for us. So Tuesday, actually, of this week, the Michigan Department of Natural Resources issued this press release on DNA testing that we conducted on 2 male cougars that were poached in that state. And again, these 2 males, they were unique individuals in our database. They were new. They didn't match a previously known individual, and they also assigned to that breeding population in the Dakotas. So this is very useful for the Michigan land managers as they're trying to think about potentially managing 4 cougars. Next slide. Here's another example from Tennessee. This animal was photographed about a year ago on a trail camera. This animal was subsequently shot, and the tissue recovered, shot by an arrow, so they didn't recover the carcass, but we were able to obtain DNA from that arrow and determine that it is a female, which is of interest because it's one of the few females that we've detected in these outside of the breeding populations. And it also, she has a genetic signature consistent again with that Black Hills, Nebraska location. Next slide. And just another example from Kentucky where they had their first cougar since the Civil War that show up and got shot. Next slide. So I just wanted to, again, make the point that these large-scale DNA databases are very important. I'm giving you the example here of cougars, but I do want to stress that we have databases like this for many, many other species that we've been using to help inform managers on movements of individuals and dispersal events. And with that, I'm going to turn the talk over to my colleague, Tommy Franklin, who's going to give you another example of large-scale sort of conservation solutions.
Thank you very much, Christy, for the introduction. And we can go ahead and move to the next slide. And once again, as Christy mentioned, I'll be talking about how we're using environmental DNA to solve conservation questions at large scales. Next slide. And so before we get too much into the background and exactly how the Genomics Center is applying environmental DNA, let's take a step back and talk about what exactly is environmental DNA. And environmental DNA is at its truest point just DNA that is shed from organisms into the aquatic system. And this could be anything from blood or feces or skin cells. Next slide. And so one of the greatest parts about environmental DNA is the sampling method. It's very quick and reliable, and it's done by essentially capturing that free-floating DNA onto a filter collected from the aquatic system of interest. Next slide. And so once we get this filter back into the lab, we have to keep in mind that this filter has the DNA from all of the organisms present in that aquatic system. And so we want to focus just on one species. So we have to have a targeted species-specific assay to essentially answer our objective. Is species X present? So we need to use that specific marker to answer that question. Next slide. And so eDNA has been gaining a lot of traction in the recent years, primarily because it's cheaper and more sensitive than traditional sampling methods, primarily electrofishing. And it's also incredibly less invasive, and as well as much faster, so you can have repeated visits with less time, less effort, and essentially less overall cost. Next slide. And so just to give you a little bit of the large scope that the Genomics Center is working on, this is just a list of some of the species that we've already completed work on. And so this ranges from exotic species to endangered species across, primarily across the western U.S. Next slide. And so while it's great to be able to get a positive or a negative detection, one thing that sets the Genomics Center apart is that we try to really look at what that detection means. So we don't want to simply just develop a tool that can be applied. We want to be able to work with you and talk with you too, so we can understand what these results really mean. So we want to be able to look at the sensitivity of what exactly a positive detection means. So how many copies of DNA do we need to have a perfect detection? And also we want to make sure that these species-specific assays we're developed are indeed specific enough so that we're not getting any false positives or false negatives in our samples. We also want to make sure that our samples are not being inhibited by any inhibition in the streams, so that we're not getting our DNA is not being bound up by other substances, so that we can overall just make sure that we're getting you the most reliable results possible. So these are just a handful of the technical studies that we've done, so that when we are working with you to design projects and understand results, we're making sure that we're taking in all of the broad-scale factors that are affecting or could affect your study. Next slide. And so while we do this, often on a local scale, we also really try to think big so we can solve these conservation questions on broad scales. Next slide. And one of the most impressive ways, and quite in our largest project, is this bull trout inventory project, in which we are validating predictions about occupancy probability of bull trout across their range in the Pacific Northwest. And while this is overall a very large-scale project, we have high resolution at local scales as well. And ultimately, the results from this project will go towards validating those results and providing more accurate models in the future that management decisions can be based off of. Next slide. And so for this bull trout inventory project, there's over 16,000 miles of streams that could be sampled. And essentially what happened is that based on climate data and habitat suitability models, over 25,000 sampling points were delineated at 1 kilometer intervals. Next slide. And in 2015, around 1,500 of these sites were sampled. And so far in 2016, over 3,500 additional sites have been sampled. And so, as you can see, we're slowly chipping away at that large number of sites. Next slide. And so while we do work at very large, broad scales, we also have high resolution to look at local conservation as well. And so this will just be a quick example of bull trout eDNA surveys in the upper Clark Fork River Basin. And the arrow is pointing to the Little Blackfoot, where bull trout have anecdotally been reported, but that area was not included in the U.S. Fish and Wildlife's critical habitat area. So we wanted to go in and work with some NGOs to sample that area to see if eDNA could detect any potential bull trout detect any bull trout in that area. Next slide. And so these red dots are sampling points within the Little Blackfoot Basin. And like I mentioned earlier, there have been anecdotal reports of bull trout, but there have not been any true collections or positive hits here in the recent years. Next slide. So the red dots here represent initial positive eDNA hits. And so we're very excited to see that we had positive eDNA detections in this stream system where we had otherwise not seen bull trout in recent years. And so once we had these positive results, we wanted to go 1 month later and just check on the repeatability of our samples to make sure that it's a pretty big deal to find these bull trout here. We wanted to really make sure that what we found was actually bull trout present in the stream system. Next slide. And so what we actually did was we repeated these results and saw that in these 5 kilometers of the little Blackfoot on the left, we repeated all of those detections. And then in addition, in Ontario Creek, we actually added a new point of detection. So this just goes to show that while we do work on such broad scales with examples of this bull trout, we also have very high resolution on local scales. And also just goes to show the repeatability and ease of sampling so that we could go back just a month later and repeat the sampling to verify our initial findings. Next slide. So a second way I'd like to talk to you all about eDNA today is using eDNA to evaluate chemical suppression of fish. Next slide. So this is an example where we worked with the state of Utah on a brook trout eradication project. And so in the fall of 2013, they started a chemical removal for brook trout. In the summer of 2013, we set up a plan to use eDNA sampling to effectively look at how well the chemical removal worked. Next slide. And so as you can see here, the purple triangles represent eDNA samples where we had a negative hit, and the gold stars represent eDNA samples where we had a positive hit for brook trout. And so this was after the first round of the chemical removal. And so as you can tell, there was still brook trout being detected in this system. So the state of Utah went back a following fall and did a second round of chemical removal for these brook trout, and they actually found a brook trout upstream of sampling point 4 where we had that positive hit. So this is just one of the great examples of where eDNA can be applied, whether you're looking to monitor sensitive species or you're looking to do eradication projects. eDNA can be seen as a very widely applicable tool. Next slide. And so while we're currently focused on about around 35 different species, 20 of these we've already completed assays for, and 16 we have been funded to develop. Next slide. One of the most exciting things, as I mentioned earlier, is that these filters contain all of the DNA from organisms in the streams. So next slide. If there is a way that we can then, instead of ask, is species X present, and instead ask what species are present, it really lends this ability to combine our expertise of eDNA and genomics that we have here at the center to look at a multi-scale monitoring tool that would save a lot of time and money on the ground, but would also give you extremely reliable results that could be applied in both an endangered species monitoring aspect or an invasive and exotic species monitoring aspect. And overall, the outcome of this would be to have an extremely applicable biodiversity inventory tool that could be used widespread, not just localized here in the Rocky Mountains, but ultimately on a national scale. And so with that, I will pass this back over to Mike Schwartz, and we can go ahead and move to the next slide.
Thank you, Tommy. So Tommy and Christy talked about how we're thinking about these problems, trying to come up with data and ask questions at local scales, but then trying to also think about this at a broad scale. And we like to do that because not only can we then turn to the state agency or the national forest or the district, and not only does that district get their question answered, is species X present, but they actually then get some context. And we believe that context is super important if we want to actually use these data to help make management decisions at multiple scales. So the last part of this talk, I would like to just spend a few minutes, and we're hoping to leave a lot of time for questions at the end. I want to spend a few minutes talking about how we're thinking about pulling this all together, the tools we're developing, the results that we're going to try to disseminate, and then if we have a chance, talk about some outreach. So if you can go to the next slide. And really what we're trying to do is we're trying to follow a model that many other people have used. We want to get to the point where we have our data disseminated online. This is from Dan Isaac's shop, and he's part of our part of the Rocky Mountain Research Station where we're housed. He got involved with us and he helped us develop some maps, and so we had this effort, this Sculpin Quest effort. We were trying to understand how many species of sculpins do we actually have in the West, what is the distribution of each species. This is a long project, actually a really fun project. It would be great to have Mike Young at some point give a seminar on this topic alone. We were actually able to probably identify 8 new species of sculpin, one of which has now been described and is now considered a new species. But here, as we try to understand the distribution of sculpins, we were putting the data was coming in from the field, from people that are interested in collecting this species or these species. We would run the DNA analysis, and we're trying to get it so we put the results online. So then the results can be online, they can be downloaded by our collaborators, who can again see not just their result and how their efforts have paid out, but also understand it into a larger context. I think this really is the model for success. It's the model that we want to move towards in the future. Next slide. And we're trying to do the same thing with bull trout. If you noticed in Tommy's presentation, and I think I'll show you in the next slide, we are trying to tie the detections. So we're trying to sample based off of a model that was produced by Dan Isaac that is very tied in with stream temperature. We're trying to, based off of that model, determine where to sample or where to advise our partners to sample, analyze the results, and then update the model. And we can do that. While we're doing that, we can put our results online. So if you go to our range-wide bull trout eDNA project website, you can start to see the beginnings of this effort. Go to the next slide. And so, again, the way that this large-scale survey is working, and I really want to emphasize this. I think it's really important, and those of you that are on the phone that have worked with us can maybe talk about this later. The National Genomics Center, we're providing kits and materials. Tommy breezed by one slide earlier that showed a sampling protocol. I think it said think big implementation. We actually have a sampling protocol that we would like to use to keep everything standard. So we have that sampling protocol online. We actually also have the kits. So if you want to work with us, we will send you a kit. That includes a pump. It's just like a library. You check it out for a period of time. You follow directions on that kit. And if you are going to use it enough, we actually ask you to buy the pumps. But then we would send all the materials. So no one has to go out there and buy materials for the, buy things in bulk that you're only going to use a small amount of. So the partners then sample. We've received some grants both from the Forest Service and external agencies then to do the genetic analysis of those samples. We then send local reports, put the information on the web, and that is again allowing this local and contextual understanding of the results. And so this is the model that we'd like to turn to in the future because I think it's a win-win for everybody. Next slide, please. And so I just want to wrap this up by emphasizing that we are going beyond just providing results on individual projects. We are going beyond just publishing. We're trying to build these tools, whether they're web-based tools for fish or in these examples. There's 2 examples here, the one on the left and the one on the right, divided by the blue line. They're different tools that we're trying to develop to help disseminate information further. And I want to show that it's not just fish, we're doing this with other things like carnivores. Let me take the example on the right first. This is a spatially explicit power analysis that we first published the basic ideas and the data in conservation biology, and this is a really nice effort conducted by Martha Ellis, who's at the University of Montana and was a postdoc here at the time. in collaboration with Colorado Parks and Wildlife scientist Jake Ivan. And basically we were saying, what kind of power do we have when we put these non-invasive genetic surveys on the ground or any type of detection survey on the ground? Do we have power to detect these rare species? But what's really important, and why I'm bringing this up here, is that we're going beyond just collecting the data. We want to have the tool that anyone can use. So in association with this paper, we actually have one other paper and then this R package that Martha Ellis put out that she put out in 2015 that allows anyone to take the data that we have or take an example data set and run it through this RSpace program to see how it works and then use their data to see what kind of power they have. We really believe in going beyond just generating data. We want to have it so that we can generate tools for everyone to use. On the left side, you'll also see another paper that was also in conservation biology, coincidentally. And this is a newer effort that actually was just published last week or maybe the week before in conservation biology. Here we've used genetic data to help understand corridors and movement of 2 different species. Once we develop our understandings of where those corridors are, we're asking, how do we decide which parcels of land are most important to either treat, purchase, or swap in an economic framework? So we're using its multi-species optimization in an economic framework so we can make very transparent solutions about where we put our limited conservation dollars. Again, I bring this example up because right now we're in the process of taking this paper, which was just published, in the optimization algorithms were just published in this paper. We're now trying to take that and operationalize it so we can have an application that's a web-based application for anyone to use. So again, the point of these last 3 slides are to show you that beyond just generating data, we want to make this data useful for all of our partners. And with that, the last slide, I just want to make sure I acknowledge that every one of these efforts is something that we couldn't undertake without our whole suite of partners. And we really have, I just want to acknowledge and appreciate that this partner network is really allowing us to look at our data in a bigger and broader context and hopefully make better solutions. Next slide, please. And so with that, I just want to thank folks for listening. Our website is listed below. And if you have questions, either you can ask them now or feel free to email me or contact us via phone or via the website. So thank you.
Great. Thank you very much, Michael and colleagues, for that.
The conference has been unmuted.
I'm unmuting all the lines at this point, and we do have a number of questions that people have already typed in the chat box here. Starting with a question, are you involved at all in cross-boundary work with the American badger between Washington and British Columbia?
No, well, that's easy. No, we're not involved with that work, but we are involved real heavily with a lynx project that's specifically designed to look at cross-boundary. Sorry, I'm hearing an echo on this, but it's specifically designed to look at cross-boundary movements, and we're also involved in fisher work to look at, again, fisher movement across the boundaries. And that's with, sorry, actually, let me just finish that. That's with Jeff Lewis of Washington, Washington State, Washington Department of Fish and Wildlife, or Fish and Wildlife, yes.
One person asked if they might be able to get a copy of the MOU that the Forest Service signed with Fish and Wildlife Service, and I suggest they follow up with you directly on that, Mike.
Yeah, send me an email. They're public documents. You can get our charter. I can send you our MOU.
Great. There was a question about the mountain lion map, going back to the map that Christy showed a couple times. There were pink dots and purple dots, and I guess there was a question as to whether those were sightings or established populations or whatever. And I'll see if I can call that map back up here, going back to that part of the presentation.
Christy, you want to
Okay,
talk about
yeah, so that map is produced by the Cougar Network, who's done a really nice job tracking sightings, and the colors of the purple and the pink referred to, they have 2 different tiers of verified sightings. And so the purple dots are some kind of physical evidence, so either a carcass or a hair or a scat. And then the pink ones are verified mostly through photographs, but they're ones that state officials or Forest Service or BLM or whatever the agency has verified that the photographic evidence, a trail camera or such, is a verified sighting. So all of those dots and the colored dots are what has been considered to be a verified presence, a verified occurrence. Does that help?
Yeah, and actually, while we have this slide up, just because you put this one up of all the maps. Yeah. We've been working with Dave Onorato with the Florida Game Commission, and Christy, do you want to mention the results? He just put these results up last week
at TWS.
At TWS. Why don't you just give a quick note to that talk and the data he shared?
Yeah. So one thing Dave has been really interested in is obviously tracking the Florida panther population through time and genetic assessment. And currently, using a combination of the Florida data as well as the reference data that we have from our other states and locations, the genetic signals of the Florida panther individuals are very distinct compared to the other populations, which I think moving forward is going to be very important as we're finding more and more of these dispersing animals pushing farther east, such that when animals show up in Georgia or Alabama or places, we can really determine whether or not that animal originated in Florida or came from the West. So.
Great, thank you. We had a question about eDNA, asking if this group is part of the eDNA and genetics community of practice.
So it depends which community of practice. I think there's many subgroups and many different, if you're talking about the Fish and Wildlife Service, they have a group of, I know they're a really nicely tied together network of biologists. And so, informally we've been working with them. In fact, I was just talking with Tommy the other day about a project that we know is going to benefit by working with that community of practice. If you're talking about the community of practice, there's a government working group. We were just at that meeting this summer. We've been tied with them since the inception. It's fantastic to be able to be in a larger network of people thinking about the same issues and how you apply these issues on the ground. Because as Tommy mentioned, it's one thing to have the gee whiz, cool, I can do this result, and it's another thing to be able to actually put it on the ground and make reliable conservation decisions based off of these data. So that's where it's been really helpful to be tied into a larger community of people thinking about how to apply these results.
Great, thank you. Question, are there any terrestrial applications for eDNA technologies at this point?
Yeah, I mean, this is a really fun conversation. I mean, it's hard. We're staring at this phone, this black box on my desk here. It's not as fun as if we were all sitting around thinking about all the really cool things you can do. There are some amazing publications out there where people have looked at watering holes and where they've been dried up and being able to detect all the suite of ungulates and carnivores that visit those watering holes. There's been some really cool stuff where people have used carrion flies. We've talked about how to use honeybees as collectors. I mean, and then going to the hive to detect rare plants. I mean, you can just let your imagination go wild, and there's just so many potential applications. We don't really know how to operationalize that at a large scale yet. We haven't seen it, and maybe I'm unaware of some studies. Maybe you guys can think of some, but yeah. And actually, there's maybe the closest. There's some really cool work out of one of the Fort Collins labs. Tony Piaggio is doing some nice work where she's looking at feral hogs. I think she's still using water there, and then also Burmese pythons, detecting those. And again, I think that's still using water, not using the soils, but it doesn't mean you can't use the soils. It's just nice to have water to help concentrate where that DNA is. Just like using a beehive would help concentrate that DNA. Using soils, the problem is it's just such a large landmass. It would be hard to know unless you could actually, come down to a discrete unit like, if you have a rub or something like that, you could probably, sample ungulates or some species like that. But again, you can let your imagination run wild, and that's really fun when we get to do that. So great question.
Great. Yeah, we have a follow-up comment here that folks in Florida are using eDNA to track pythons using soil. So that's interesting information there. Thank you. Question from folks in the Forest Service. Has there been much conversation on how these tools and approaches can be used to support the forest planning process?
Yes. I mean, that's one of the reasons that I think we exist is that I think, all of our legal mandates, whether it's NEPA and the planning rule or meeting in ESA regulations or consultation regulations, I mean, we've seen our data used many times in each of those components. For the planning rule, for instance, I think there's focal species and species of concern. So species of concern have focal species have a monitoring component to it, and we think some of these species of concern, some of these focal species, the only way to actively monitor them across time are going to be using either non-invasive samples like we've done in the past, where we've been able to look for, over long time horizons of how populations change of that focal species, or using these new eDNA approaches if it's an aquatic species. And again, I think those are going to be the only solutions because in the Forest Service, oftentimes these species exist at such broad scales, they pose such immense challenges. And actually what we tend to find is that when we actually apply these tools, when we apply non-invasive tools, whether it's eDNA or non-invasive genetic sampling, to try to understand the distribution of species, we often find that things tend to be a little bit overmapped at the beginning. So when we think species are in maybe more places than they are, and actually, or they actually show up in places where you don't know that they are. And that's really helped our forest planners, or at least we've heard from them in the past, that that's really helped planning because we're able to, one, remove constraints where they aren't, and two, place conservation dollars where they are.
Great, thank you very much. We have several folks who are interested in potentially collaborating with you, and the question is how much capacity do you have to take on new projects? How much are you already working in the U.S., and how would folks go about initiating and possibly even jointly funding a project? So a related cluster of questions there about how can folks follow up and work with you, and I just put your email out there to the group as well. So.
Yeah, thank you. This has been a real interesting issue for us. The growth has been astronomical for us. We really are prioritizing some of our partnerships, and really the things that we want to get involved with are where we can test this new model out, where we're bringing, at the first level, we want to be working with people where they can potentially send us have somebody that's a point person that can work with us, and that person maybe comes out here for a little bit of time, really understands the method, helps us understand the question, because I think we can ask smarter questions. And often our limitation is personnel and people time and trying to understand the question and getting that part right. And vice versa, we want to see the results go to ground. We love it. There's nothing better for us than when we see how our results are being applied. So yes, our previous model actually was enormously successful, but I felt like we were still missing something. So anyone that can collaborate with us in that capacity, they're going to be able to just walk right in the door. At least that's how I feel about it, because I want to make sure we understand the question best and the results get to ground fast. Having said that, we also do some fairly standard collaborations, and we're not at capacity. We're trying to understand where our capacity is at. Unfortunately, we're scientists and not business folks, so you have to bear with us a little bit. We still feel like we can be taking on some large projects, and we're interested in taking on large-scale projects. Those actually tend to be a little easier on us than a lot of small projects. So if it's an eDNA project, I would encourage you to contact Tommy Franklin, and I'll send out his email. I can send out his email later, or just go to just type in National Genomics Center or Forest Service or my name, and you can get to our website, and his email should be on there, hopefully. Tommy just started a few months ago. As I mentioned, Kelly Kareem, who a lot of you have worked with, she's now our tribal liaison position, a more traditional type postdoc. So you can either email myself, you can email Tommy, you can go to our website. We're interested in working with you, and we may already have some of these, some markers developed that you can apply immediately.
Great, thank you. And I've also posted the phone number for Mike's lab as well, but certainly the email, the website, Any way you'd like to contact, please feel free to follow up. We hope that this webinar and these contacts lead to some fruitful collaborations. We do have a question here about has the lab considered working on sculpin species in the eastern United States?
Oh, well, we haven't actually tackled sculpins in the east, but we actually just recently, I was just in D.C. a couple weeks ago, and there's a lot of interest in exploring the potential of moving to some of the aquatic systems in the east. And so we're going to start exploring that in a very calculated way, meaning we don't want to just go at it by whoever can come here first. We'd like to actually think what the biggest need is and who can we partner with to develop some meaningful projects together. And actually, we're real fortunate that Tommy, we hired him because he was working on hellbenders on the East Coast in, let's see, North Carolina and just North Carolina?
Tennessee and Virginia.
Tennessee, North Carolina, Tennessee, and Virginia for his master's work. So he has some familiarity with the Southeast, but we really do want to move into some of these other places where we haven't before, where the Forest Service has asked us to move into. But again, we want to make sure we're doing it in a strategic way where other people aren't already there, or where we can actually apply some of our tools, especially as we're doing this kind of metagenomics. Because obviously species richness is enormous in the Southeast compared to, we're barren up here compared to the Southeast, right? So I think it's really important to make sure we can bring the right tools to the right place with the right partnerships.
Yeah, and speaking of partnerships, I've got some comments here saying that the Fish and Wildlife Service has a Fish Technology Center in Georgia that's doing some eDNA work as well. So there may be fruitful opportunities for collaboration there. And again, I'd encourage anyone on the line to please feel free to follow up with Mike and his colleagues if you have specific questions, ideas for projects, and so forth. And that exhausts our questions that we have in the chat box at the moment. So we have a few minutes left before we need to close the line out. So if there are any folks on the line who have questions that have not yet been raised, or anything you'd like to ask Mike and his colleagues, please feel free to do so at this point.
And actually, while you're waiting for those questions, I just want to make sure I acknowledge that the beauty of this MOU is that we have with Fish and Wildlife Services to work together with these fish technology centers. There are some tremendous researchers, and they have them spread throughout the country, that are doing some great work in those centers. And so we just really believe that there's too much work to do out there, and it's such a waste when people are sampling the same points and when we don't gain the synergy. And so I'm really excited about that collaboration and seeing where we can take that.
Great. Good. Any other questions from folks on the line?
Hi, this is Ann Tim in Baltimore, Maryland, and I asked the sculpin question. So when you were talking about kind of strategically partnering with a group in the East, how do you maybe envision that going about that?
I have some ideas. I can give you a good answer, but I'm going to put John Roethlisberger on the spot here, because he's been involved in these conversations of how the Forest Service could potentially think about prioritization of work throughout the country. So if John wants to jump in, I think he's thought about this a lot, and it might be more poised to answer this.
Okay.
Sure. Thanks, Mike. Hi, this is John. Hi, Ann. Great question, and I agree that we're looking to expand the work in the East in the near future. And like Mike said, there's just so much to be done that we need to be really strategic about what species to focus on, and the rationale for focusing on those species, in particular as we develop eDNA markers for those species. One of the things that we've been talking about here in the Forest Service National Office is convening a small work group that would include representatives from national forests throughout the nation, and from partner agencies and organizations like the Fish and Wildlife Service and the USGS, and university partners that are working on eDNA issues, and identifying what species we have covered with markers right now, and what are some of the key species that may be highly valuable to national forests for their broad-scale monitoring requirements under the 2012 forest planning rule, and looking to see where there are gaps, and whether it be geographically or taxonomically, and coming up with a prioritization to filling those and a plan for which labs, which work groups will start developing those markers so that we can do it in a coordinated fashion. So it's a long answer to your question, but I think the upshot is that in the near future we'll be trying to convene this group, and we certainly look for expressions of interest for people that want to be part of that.
Thanks, John.
This is Cindy Williams. I'm with the Fish and Wildlife Service. And one of the places that I think we should start working together on some of this stuff, especially when we're talking about trying to prioritize species, would be to start with the mega petition that we have settlement agreement on with the Center for Biological Diversity to make listing decisions on over 600 species, 457 of which are in the southeast. Most of those are aquatic. So that's kind of how we're looking at some of that stuff. But that'd be a good place to potentially start, just a suggestion.
Yeah. This is John. Cindy, I think that's a great comment. And Seth Mott of U.S. Fish and Wildlife Service with the Science Applications branch is part of the group that helps to advise the center. And he brought up the same point with the mega petition. So I think that's one of the key things that we need to be looking at.
Great. Any other questions from folks on the line? Well, if not, I'd like to thank all of the presenters, Mike and his colleagues, for Tommy and Christy for the really excellent presentations this afternoon. Thank all of you for your work in this up-and-coming area that's of great interest to state and federal agencies alike. Thank you for taking the time to share your work with us and for enlightening us about the work that's going on at the center and some of the great new opportunities for collaboration and cooperation among colleagues. Thank you especially to the U.S. Forest Service for co-presenting this with the Association of Fish and Wildlife Agencies. And before we finally sign off, I'd like to turn it over to my colleague John Roethlisberger for any last words from the Forest Service.
Great. Thanks, Jonathan. And I'd just like to second your comments about the excellent presentations. Really appreciate Mike and Christy and Tommy for what they shared, and would like to thank everyone else for participating in today's webinar. You can tell from the questions that the presentation stimulated a lot of great thoughts about opportunities to work together to address fish and wildlife conservation challenges using genetic and genomic data. And I encourage you, as you've encouraged several times, to follow up on those stimulating ideas. Please feel free to reach out to any of today's presenters, to myself or to Jonathan, if we can help make connections so that we can make sure that we follow through on some of these ideas. Finally, I would just like to thank Jonathan Maudsley and the Association of Fish and Wildlife Agencies for organizing and hosting this webinar series. In fact, we have another webinar in the series coming up in just a few weeks. That will be on Monday, November 14th, and that presentation will feature Dr. Katie Moriarty. Katie is actually closely involved with the National Genomics Center, so this will be a nice follow-on. She will be talking about marten genetics and conservation, the insights into marten conservation that have come about through some of the genetic work that she's worked with the center on. So we look forward to that presentation. We, of course, always appreciate feedback about these webinars, and we're interested in any suggestions that you might have for improving them, and also suggestions for future topics. So please feel free to share those with Jonathan or myself. And again, thank you very much for your time today and for all the great participation.
Great. Thank you very much, John, and thank you all for participating in today's webinar. We're going to turn off the recording and end the webinar at this point. Thank you very much. Have a great afternoon.